I'm Lauren Bunnell, and I will be discussing my research in investigating the role of subtelomeric CTCF in telomere stability. Human cells have telomeres, which are repetitive units of nucleotides that act as protective caps on the ends of our DNA strands. Every time a cell divides, a small piece of DNA gets chopped off the ends of our genome, so the telomeres are there so that they are what is shortened rather than important genetic information. And a large part of cellular aging has to do with the rate at which these telomeres shorten. Cancer cells have the ability to increase the length of their telomeres, which allows those cells to be extra protected, and as they build up and form, this can lead to the formation of tumors and cancer. So the overall purpose of this research is to further investigate certain properties of telomeres so that we can better understand their role in cancer cells. CTCF is a multifunctional protein that binds all over our genome, but this research will be studying the CTCF proteins that bind directly near the telomeres and control the transcription of the telomeres. Transcription of telomeres forms telomere repeat containing RNA, or in other words, TERA, which has been shown to play a very important role in successful telomere replication and stability of telomeres. And the functions of CTCF and TERA are not entirely understood at this point, so this research will be building on the foundation of the understanding of those structures. It's important to know that CTCF plays a key role in transcribing TERA, and when cells lack the appropriate CTCF binding site, studies done by Dr. Beischland and her team discovered that the cells were much more sensitive to stressful conditions during replication, which led to the formation of ultrafine anaphase bridges. And anaphase bridges are sister chromatids that are still linked together and are unable to successfully separate from each other during mitosis. And these bridges can lead to instability and mutation and even cell death because the divided cells either have more chromosomes than they need or less, or even none at all. However, when TERA transcription was increased, it protected the cells from the formation of these anaphase bridges. And this is important to know because it shows that you can rescue transcription in CTCF mutated cells to stop the formation of the bridges by increasing TERA transcription. The objective of this research is to learn how the presence or absence of subtelomere binding of CTCF affects the DNA replication and cell division of telomeres. I will be focusing on a specific phenomenon in cells that acts to promote transcription to avoid the formation of anaphase bridges, which will ultimately rescue the DNA and allow for successful cell division. A 2011 study by Jasket Leoff elaborated on telomere reactivation and showed that a cell can attempt to complete small amounts of replication during mitosis, which allows the cell to divide without mutation caused by anaphase bridges because the bridges wouldn't form. And I will be determining if CTCF binding and TERA transcription are truly necessary for the rescue of DNA replication at the telomeres during mitosis. To do this, I will be culturing human colon cancer cells, some of which will be mutated to block CTCF from binding near the telomere, and others which will be non-mutated wild-type cells to serve as a control. Both types of cells will be studied under conditions of replication stress so that we can observe how the telomeres are behaving during mitosis. In the mutated cells, TERA will not be activated because the protein is absent, so the telomeres will be attempting to replicate, but they won't be able to do so normally, which will result in either complete absence of a telomere or maybe a smaller than normal telomere depending on how the replication occurs. And then after we expose the cells to stress, they will be released into mitosis with EDU, which is a signal that detects replication in cells. So then the cells will be stopped as they are in metaphase so that the chromosomes are condensed, which will allow us to be able to image them clearly. At this point, when they're in metaphase, I will add potassium chloride to cause the cells to swell, and I will drop them from a height onto slides, and the impact will cause the cells to burst and the chromosomes will spread over the slide and form what is called a metaphase spread, which is how we will observe them for their immunofluorescent properties. The EDU signal will show up at the end of a chromosome as a little red signal, and the signal will be present in the wild-type cells because replication is completed during mitosis, but we're predicting that in the chromosomes of the mutated cells, they will have decreased red signals, meaning that replication was not occurring as much or maybe not at all. The characteristics of what is seen will show if the cell was trying to rescue its DNA during mitosis or not, and a student t-test will be used to determine if there's a statistical significant difference in the number of signals seen in the wild type and the mutated cells. Overall, this research is intended to explore all of the ways that the CTCF binding site contributes to telomeres so that we can better understand the dynamics of telomere pathways in aging cells and therefore in cancer cells. We know that CTCF is always present in cancer cells, so studying how it functions, we can potentially find a way to find therapeutic targets to prevent cancer formation. And the more we can understand how it functions, the more we can understand how to control cancer pathways related to cellular aging.